Hello guys, let's talk about the enzymes today. As we all know, enzymes, they're the biological catalysts that are found in the living organism. The two ways we can define the enzymes, one which can enhance the rate of reaction. Secondly, on the basis of the kind of molecule they're made up of. Most of the enzymes are made up of proteins, but there are certain enzymes which are RNA molecule. They're also called as ribozymes. That means basically the active site of the enzyme lies in the RNA molecule. They may have a supporting proteins all around. For example, the peptidyl transferase or the splicing factors. They do have the proteins, but mainly the reaction is going to be catalyzed. All the transcriptification reaction in the splicing process is catalyzed by the RNA molecule, snRNAs. So, a very warm welcome from the Spice of Your Biology, Sandeep here. Today's topic is going to be enzyme, as I told you in the beginning. Let's talk about it. So, why we require enzyme in the biological system? So, there are the few things which is very important we need to understand. First, it will enhance the rate of reaction several times. To be precise, around 10 power 6 to 10 power 12 times greater than a reaction which is non-catalyzed by the enzyme. Second, it requires a milder condition. Usually the temperature is going to be ambient. It varies though from one organism to the other. Pressure is going to be optimal, and the pH that could vary. Normally, most of the enzymes, they have a pH in the neutral range or slightly alkaline, but it may vary as per the requirement. But the conditions are not going to be very harsh, except that, some of the extremophiles or some extremozymes, they derive from the extremophiles. Third, they have a greater reaction specificity. They're very specific to their substrate. One enzyme may have more than one substrate, and some they have just one. There are the many ways they can vary. Even some of them, they can be stereospecific. So normally the enzyme usually of the range of the protein amino acid is going to be in the 1,000, and it could be less than or more than that as well. The fourth thing which is very important, once we have the enzyme, we have a total control over it. Under all the conditions, we can regulate it many ways, production wise, or maybe the allosteric enzyme, maybe other feedback inhibitions, we have a control over it, or it can be controlled at the gene level as well. And hormones do play very important role in order to control it. Look at the some of the enzymes you can see in this table, and look at if the reaction which is carried out by the carbonic anhydrase, which is one of the fastest enzyme, which convert carbonic acid into the H plus and bicarbonate. If the reaction is not carried out, it's going to be 1.3 into 10 power minus 1 per second. But if it is the enzymatic reaction, it's going to be 1 to 10 power 6. Just look at enhancement time is going to be around 10 power 7.7 to 10 power 6. Look at the last one, staphylonuclease. The rate of reaction is going to be extremely slow. It's going to be 1.7 and 10 power minus 13. And if it is enzymatic, it's going to be 95 per second. Here, the enhancement rate is going to be 5.6 and 10 power 14. So, as I told you in the beginning, they could enhance the rate of reaction quite a lot. Now, how exactly the enzymes are given the names and their classification. So, entire enzymes on this earth we can put into the 6 categories where they perform a particular reaction based on that. For example, the first one is oxidoreductase. Here, the oxidation or the reduction reaction will take place. Second is the transferase, where some functional group is going to move from one place to another. Third one is the hydrolysis, which will be involved in the hydrolysis. Lyases, which is going to make the double bond. Isomerases, one isomer will be converted to the other. Ligase, it is the joining of the reactions. So this is a little more detail, oxidoreductase. and the transferases, hydrolases, lyases, isomerases, and the ligases. So if you just see the reaction and the few examples of that oxidoreductase, I told you they're going to catalyze the redox reaction. And best example you can see over here is the LDH, lactase dehydrogenase, or any kind of dehydrogenases. Look at over here, the lactate which is going to be formed, and this requires NAD plus. So here the pyruvate can be formed, and this is NADH which is going to be formed. So this reaction, if you see the reversible, most of the enzymatic reactions are going to be reversible, but whereas wherever it has irreversible reaction, that will always be playing important role in the regulation part. Now the transferases, you can see over here, one group is going to be transferred. Like for example, here the enzyme that is shown over is a PNMT, and this is a norepinephrine that is getting converted to the epinephrine. Here, one methyl group donor is going to be there, and this is going to transfer the methyl group. Even all the kinases, you can see the DNA kinases or the protein kinases, they will always transfer the phosphate group onto the group. Then we have the hydrolases, which is involved in the hydrolysis. So most of the biopolymers, like you see the proteins, or you see the carbohydrates, or you can see the DNA, RNA, even lipid, which we cannot keep in the category of the polymer because they do not have that higher molecular weight, but they do also form the ester bond, and that also can be hydrolyzed easily. For example, the example shown over here is of the lipases, which is going to work on the triglyceride. Here you can see the 3 water molecule is required, and this is how the glycerol and fatty acid is going to be separated. This plays important role in the fat digestion. Lyses, as I told you, they're going to be involved in either making the double bond, or they're going to form the double bond. So lyses catalyze the removal of the group to form the double bond, or they reverse back the double bond. So all the decarboxylases and the synthesis, for example, the ATP synthase, they are going to be part of the lysis. Isomerases, you can see over here, the best example shown over here, the epimerases and the mutases, and many isomerases are there. You can see the phosphoglycerate mutases. You can see this phosphate group which is present on the carbon number 3 can be transferred on the carbon number 2. So 3 phosphoglycerate to 2 phosphoglycerate. Ligages. Ligase means it is going to join together. So ligages catalyze a reaction in which carbon-carbon bond, carbon-sulfur bond, carbon-oxygen bond, and carbon-nitrogen bond is either made or it's going to be broken, because it is having a reversible reaction. So one way it is going to form, another way it can be broken as well. Now come to the nomenclature. So if you look at in the most of the cases, the enzymes, they are given the name based on the product or the substrate they have, and they're going to end up with the ASE, ASE. So common name for the hydrolysis is derived from the substrate. For example, urea. One which breaks the urea into water and ammonia is going to be ureases, lactoses. Lactose is going to be broken down into the glucose and the galactose with the help of the enzyme because of lactase. Some of the other enzyme where some other reaction is going to be catalyzed, especially dehydrogenase, means is a removal of the hydrogen. That means this reaction is going to carry out for the oxidation purpose. Similarly, the pyruvate decarboxylase, which is going to remove the carboxylic group, or CO2 removal is going to take place. And some of the trivial or the historical names are also given. For example, the catalyzes, pepsin, chymotrypsin, trypsin. These are having the historical names. Now, one question that comes in our mind is that how exactly the enzymes are going to be playing important role in enhancing the rate of reaction. Let me first tell you that enzyme will not be involved or not in the, neither consume in the process of reactions. They will catalyze the reaction and come free. If they are being consumed in the reaction, they will not be enzyme. The one thing. Second thing is that, they do all this just by reducing the activation energy. Activation energy is, one energy that requires to carry out the reaction. So that lowering of the activation energy will always decrease the reaction time. Hence, the rate of reaction is going to be faster. For example, if you see this, any reaction which is happening, if you see this A, and B. So if you want to try out the equilibrium constant, so B and A is going to be the product. Now, if you see over here, this is the reaction which is uncatalyzed one. So this, the peak over here is going to be the activation energy. So if the two substrate or reactant A and B reacting together to give the product C and D, that means in order to have this reaction, the energy level should go up to this level, this peak. This is called activation energy. If it is catalyzed by the enzyme, look at this. There'll be lowering of the activation energy, hence the reaction will move quickly and at ease. Now, another thing which is very important here is that the rate of reaction versus the substrate concentration. Normally, if the reaction is uncatalyzed, you get a straight line. As you can increase on the substrate concentration, the rate of reaction will increase. Whereas if you see in the enzymatic reaction is going to be hyperbole. Though I will not say like it will work for all the enzymes. This is for those enzymes which follows the Michaelis-Menten equation. Suppose an enzyme is allosteric enzyme. So this is not going to be there. The graph is going to be different, that they will, follow the sigmoidal curve or they will follow the monad kinetics. So that you have to be very careful about. But we all know most of the enzymes are not allosteric enzymes. There are only few enzymes which are the allosteric enzyme. So here, one more thing, like if you see over here, in the beginning, until it reaches the Vmax, it is dependent on the substrate concentration. Once the Vmax is arrived or reached, further increase in the substrate will not increase the rate of reaction. That means here, till the Vmax reaches, it follows the first order reaction. And once like the Vmax achieved, after that, it will follow the zero order reaction. So overall, the enzymatic reactions are pseudo order reaction, which is a mixture of the first order and the zeroth order reaction. So this is a better diagram to show you that. So one which is shown in the red is going to have the catalyzed one, and one which is shown in the blue is going to have the uncatalyzed. So the energy difference over here, that is a delta G, which is the going to be saved by the enzymatic reaction. Now, how exactly the reaction is going to happen? So if the reaction is reversible, if you see over here, so enzyme and substrate, they combine together, they form the enzyme substrate complex. Then they will go to the one energy state, transition state, ES, where the energy is going to be maximum. And this will lead to the conversion. In transition state is one where the new bond is being formed and the old bond is going to be broken. This is the halfway or midway process where it can go either way. So if it is going to be stabilized better, product formation is going to be more. So then the EP, enzyme and product formation occur in the complex form, then the product is going to be released. This is how the reaction is going to happen. So overall is a 4-step process, and that is well described by the enzyme kinetics given by the Michaelis-Menten. Now, if you see over here, if you just see carefully, this is the substrate. Let me tell you first. So the native form of the protein is going to be very, very important because it has a very small section which will arise due to the folding of the protein. This is also called the active center or the catalytic center of the enzyme, which is very, very, the group is very, very important for the binding of the substrate. You can see over here, this is just diagrammatic representation, but you see, this is the one which is going to fit over here, and this is the one which is going to fit over here. And the entire substrate is going to sit over here. So it is a chemical reaction, rather I should say the biochemical reaction, So it will always be having certain interactions. Mainly those interactions are going to be non-covalent interactions. Rarely you have some covalent interactions happening, because covalent bond is going to require a lot of energy to be broken. Hence the reaction rate is going to be slowed down, or the energy consumption is going to be more. So more than often in this process, this reaction is going to be always, the bonds are going to be non-covalent bond. Now, how exactly this complex is going to be formed? Let's talk about it. So the part of the enzyme which is combining with the substrate is going to be active site. I told you the small pocket, you can say. Active site will always be having, such a structure where the R groups are protruding. And these are the catalytic groups which will have the interactions with the incoming substrate. Shape of the active site is going to be complementary to the shape of the substrate as per the lock and key model. Enzyme attracts and holds the substrate using the weak non-covalent interactions I told you in the beginning. And sometimes this conformation of the active site determines the specificity of the enzyme as well. I'm just talking about the stereospecific enzyme. Let's talk about the few models which has been put forth to explain this. One is a lock and key model. It's one of the oldest model. Emil Fischer was the one who had given this. Here, the lock is of your active site of the enzyme, and the substrate is just the keys which can fit in. So you can see the substrate can easily fit in the active site of the enzyme. Another model is called as induced fit model. So this came late, by the Koshland, and this is the one. It says that when the substrate comes, there'll be some conformational change will be induced in the enzyme to fit in with the substrate. Hence, the name is given induced fit model. Because all the aspects of the enzyme kinetics cannot be explained by the lock and key model. You require the induced fit model. Now the question is, which one is better? I would say none. Both are required to explain the enzyme kinetics. Otherwise, you won't be able to explain this, especially the steady state and transition state theory, which can easily be explained by the enzyme kinetics. Now the specificity. So the few ways we'll talk about it. For enzyme, the substrate to react, surface of each must be complementary. The one thing means one is positively charged and there's going to be negatively charged, or one is having the regions which are hydrophobic, so another must have the hydrophobic in order to have the hydrophobic interactions. So enzyme specificity, ability of an enzyme to bind only one or very few substrate, thereby catalyzing only a single reaction. If you just look at this two reaction here, urea is going to react with the water to give you the CO2 and ammonia. This is the urease, which is there. And the methyl urea, just one substitution of the methyl group, there'll be no reaction over here. So it is very, very specific to the substrate. So that shows the high degree of specificity in this particular reaction, not in all the reactions. So the few things which we need to talk about once we discuss enzyme specificity. First, absolute enzyme reacts with only one substrate. If it is absolute specificity, maybe the group enzyme catalyzed reaction involving any molecule with the same functional group. For example, you can see the hexokinases. Linkage, enzyme catalyze the formation of or the breakup of the only certain category or type of the bond. And the fourth one, stereochemical, enzyme just requires the only one of the two enantiomer. For example, most of the carbohydrate which is found in the nature is in the D form, capital D I'm talking about. Whereas all the amino acids is going to be L form, which are present naturally. So amino acids are required in the L form and the carbohydrates are required in the capital D form. This shows because the enzyme is going to be specific to these two enantiomers only, not the other way around. For example, the L sugar or L carbohydrate is very difficult to be digested because we do not have enzyme specific to it. This is how you can see the specificity. And if you change the group or the positioning, So it won't be able to bind. This is the green surfaces of the enzyme. These are the pockets where these groups are going to set in. And if you change the structures, this group, if it goes like, on the left and the left part goes right, it will not be able to fit in properly. So this is, what is called stereospecificity. Now the transition state and the product formation. How does the enzyme promote a faster chemical reaction? I told you in the beginning, the two things which is very important in this regard is one, the first part is that, how exactly it lowers the activation energy. Second, it fits in with the substrate and converts into the transition state, which is going to be stabilized for a while. And then the reaction is going to take place. So substrate interacts with the enzyme, its shape changes, and the new shape is less energetically stable. And this transition stage is a feature of both the substrate and the product, and falls apart to yield the product which dissociate from the enzyme, provided the conditions are such which promotes the product formation. If it promotes the substrate formation, it might go other way around. Now, what are the possible changes which occurs during the reaction? Just look at this diagram which says all. If you see the enzyme and substrates coming together, look at over here. So this is the ES, enzyme substrate complex. Here the transition state, you can see this is a little protruding. That is, putting a force on this. And this bond is, now little more tends to be broken. And here it is going to be broken and the product is going to be released. The second thing is that the enzyme might, bring the two reacting groups. For example, if these two reacting groups are, there, if they are on the other side, it will be very difficult to come across. So they also orient the functional group to come together to have a reaction. Then this reaction is going to happen easily. Look at this. So enzyme and substrate coming, the red one is showing which is going to react. So if they are far, so reaction, the possibility of having reaction is going to be lesser and time it requires is going to be more. So what enzyme is going to do is that it is going to, reorient them and bring the functional groups together. And then the bond formation is going to be facilitated and the product is going to be formed. Third, enzyme might modify the pH of the microenvironment, donating or accepting the H+ ion. So that is another way. And in this process, there'll be some other groups which can help it out. For example, the cofactors and the coenzyme. Active enzyme, it could be the holoenzyme. The protein part we call as a coenzyme. And the non-protein prosthetic group is called as cofactor. Cofactor is umbrella word, which can have, the many things into it. Could be the metal ion. It could be organic compound. It could be organometallic compound. So depending upon the factor. Usually the one which is tightly bind with the enzyme is called as prosthetic group, and one which is loosely attached with this is going to be called as coenzyme. So all is going to be kept in the category of the cofactor. So what are the factors which are responsible for the catalytic efficiency of the enzyme? One is a proximity and orientation. I told you all the reacting groups is going to be brought together. Orbital steering, stereo population, control, substrate anchoring, rack or distortion mechanism, and coenzyme already I told you. These are the organic molecule which is going to have the weaker bond. There's a few examples of the enzymes shown over here. Basically, they are the vitamins. And the effect of the pH. So you get always the bell form structure, pH and temperature. Those are the two factors which we'll discuss, in the next lecture, whenever we'll get the. Thanks for hearing me. Hopefully you enjoyed this lecture. Thanks a lot.